Hi everyone. In this lesson, we're going to explore one of the most important biological processes. It's called protein synthesis. So, protein synthesis is basically how your DNA turns into your traits, your structure, your enzyme, literally everything. So, let's get started. First of all, let's just clarify what is this word or this huge word, central dogma, because a lot of people hear it and they get confused. So, central dogma it states that everything starts with DNA, and we know that, right? If me being tall or short, it's because of my DNA. But central dogma actually clarifies it more in details. It states that it doesn't actually just starts with DNA and ends with DNA. It actually starts with DNA, go to RNA, and from that RNA we're going to make proteins. So DNA doesn't do it all alone by itself. It needs help from RNA. And then this RNA will also help us to make those proteins. And those proteins that are actually responsible for basically our traits. Think of it like the DNA is the blueprint, RNA is the messenger, someone in between, and the proteins are the workers that actually make things happen in your body. And also genes. A lot of people like they don't still know what is a gene. So the gene is a small section of DNA that codes for a specific protein. So every protein has its own specific gene, and that gene has codes that code, or if they being translated, they will be making a protein. So those proteins that gives us all those traits. It's not the genes. The genes are just being used to make proteins, and then the proteins will be responsible for giving us all our traits like eye color, dimples, if we're gonna have dimples or not, even our cells functions. Next is gonna be the protein synthesis. When we're talking about synthesis, we are making something. Protein synthesis is basically the process of making proteins. It goes in two very important steps. The first one is the transcription, where the DNA is being copied into a messenger RNA, and the translation where that message, that exact message, is being used to build a protein. It's like a recipe. So just to note that transcription happens inside the nucleus where the DNA is, and translation happens outside in the cytoplasm. Or if we're being more specific, it happens inside the ribosomes. We're going to get more into details later. So let's zoom in a little bit on the transcription. It happens inside the nucleus. The DNA is first of all unwinds or unzipped at the gene that we want to make a copy of it. So who does that? Is this guy called the RNA polymerase. This one basically reads the DNA and creates a matching RNA strand for it. And this mRNA carries the messenger or the message. He is the messenger itself. And then we'll carry this message out of the nucleus into the cytoplasm. And then he will find his friends. So if you can see here, we have a modeling of a model of a transcription. So just to remember, if the DNA strand reads ACT at the beginning from the left to the right, the transcripted strand is going to be UGA. Just if you notice that in the RNA we don't have a T, we don't have thymine, we have the uracil U. So for DNA we have four bases A, T, C, G. But for the RNA we have AUCG. We don't have the thymine. So just remember this, very important. And here the newly made RNA will go basically outside of the nucleus, and we're gonna have our own message. So just to remember, we're trying to make a protein. And now we already have the language or the message that we need, and then we need to translate it. So they should go outside of the nucleus and find those guys called ribosomes. Ribosomes are the ones that actually go in to help. So here we have the codons. The mRNA will have codons. Every three bases, they make a codon. For example, we have the codon UAC. How do we translate it? Well, basically we use this wheel or the codon wheel, or also sometimes people use the codon chart. It's like a table, but this one is more easier. So how we do it? We always go from the center and going outside, from inside to the outside. If you notice that inside we have ACUG, we don't have a T. So you guessed it right. We are talking about RNA. We are using mRNA. After transcription, forget about DNA. We're only using mRNA, right? So we always go from left to the right. Our mRNA is reading UAC. First of all, we have to find U in the first inner circle, U. And then the second circle, we're going to find A. U, A, and then the left one is C. So we have to find C. Is going to be the tyrosine, right? You can do the second one. G, C, and U. You find the G in the very inner circle. The second circle is going to be, you go on to find C, and the third one you're going to find U. Let's see the results. Here are the results. So our amino acids are going to be the tyrosine and the alanine, right? Very great. So here we have the RNAs. We have to clarify some of those RNAs and what they do. So the mRNA we already know. It copies the DNA, take that message from the DNA inside the nucleus and take it to the ribosomes. rRNA, it makes up the ribosomes themselves. And the tRNA is basically matching the anticodon of itself to the codon on the mRNA to bring a very correct amino acid, because it cannot just be done randomly. Every amino acid should be matched to its exact codon on the mRNA. Otherwise, there will be a problem there that will maybe like developed into a mutation or a mistake. So next one is going to be translation. So the translation, if we zoom in inside this process, we're going from the mRNA to the protein. So it happens in the cytoplasm, exactly specifically inside the ribosomes. Who's doing this work? We have three guys: mRNA, ribosomes, or also we call them the rRNA, ribosomal RNA, and the transfer RNA, the tRNA. The first thing is the mRNA will find the small unit of the ribosome, binds to it, and then this mRNA codons are going to be read by the tRNA that already has codons. Here you can see them. It has mRNA has the codons and the tRNA has the anticodons. So these ones should be matched. Otherwise, there will be no bond and then there will be no translation in the first place, right? And then the next one is going to be the amino acid linked together. As you can see here, amino acid is going to be linked together and then making a chain of amino acids. Those chains of amino acids that we actually call a protein. So if you understand what we were talking about now, let's do a practice. You can do it by yourself here, and then This one is already solved, but you can also try to find the reasoning behind it and you can understand it very well. So the first one, we have the DNA strand that we're gonna first do the transcription for, and then from that mRNA we're going to find the exact protein chain or the amino acid chain that going to basically make a very nice or very long protein, right? So to sum it up, the DNA gives the instructions, mRNA carries the message, ribosomal RNA or ribosomes themselves plus the tRNA, they will help or make the protein. And then this whole process is basically, if you think about it, it's happening right now inside your body every second. And that's what actually making you alive and basically keeping you alive and functioning. Right, so thank you very much for watching this video. And if you like it, subscribe, comments, and like for more videos to come. Bye bye.